Hello everyone. So today we are going to talk about the structure of coronavirus, also known as SARS-CoV-2. So as you are seeing in this image, this is the structure of coronavirus, and it is around 120 nanometer in size. It ranges from 50 to 200 nanometer, but most of them are around 120 nanometer. Now this black colored strip that you are seeing, okay, this shows us the viral envelope, the envelope of all the viruses contain an envelope around them, and this is the envelope of coronavirus, and this envelope is made up of lipid bilayer. The next thing we need to remember is the three structural protein that are present in the membrane of the coronavirus. These are the M protein, means the membrane protein, the E protein, the envelope protein, and the most important and the uh, crucial protein, that is the spike or the S protein. Now these three are the structural protein, but the important function in determining the size and the shape, that is the structure of coronavirus, is through the M and the E protein, the membrane and the envelope protein, whereas the spike protein or the S protein is the most important protein in the sense because it is responsible for the fusion of virus to the host cell, as well as it has the receptor binding domain to attach to the receptors. Now let's understand a bit more about the S protein, which are the most important. So now we will try to understand a bit about S protein. Now this S protein is made up of trimer of the S proteins, means each unit of S spike that is present over the membrane of the SARS-CoV-2 has a trimer of S protein. Now this trimer of S protein is made up of two subunit, that is S1 as well as S2. Now when the coronavirus is active, there are two S2 units that are attached to three S1 units. These S1 units are the heads and the S2 units are stem. As you are seeing in this image, this is the S2 part that is acting as stem and is anchored over the envelope of virus, whereas S1 act as head, which contain the receptor binding domain that is going to bind to the ACE2 receptors. Okay, so this is what we needed to know about the S protein. That S protein is the receptor contains the site for receptor binding domain, and it also helps in the fusion of the two cells, that is the coronavirus as well as the host cell. Now each coronavirus contain approximate 74 spike proteins, and this S protein or the spike protein is responsible for the characteristic crown-like appearance of coronavirus on electron microscopy. The ratio of the E, S and M protein is as follows, and the nucleocapsid inside contains a positive sense single-stranded RNA genome. Now this differentiates a coronavirus from influenza, which contains a segmented genome. Although it is not segmented, still it is showing a lot of mutations, and it is positive sense. It means that it is directed 5 prime to 3 prime, and it can directly replicate, whereas in the case if it were a negative sense RNA, it first undergoes change into 5 to 3 prime and then undergoes the translation, but here in this case it does not happen so this is what we needed to know about the structure of coronavirus. One more thing is that in some of the strains of coronavirus, there is another protein, that is hemagglutinin esterase. Now this protein is responsible for attachment and detachment of coronavirus over the host cell. So this is what we needed to know about the structure of coronavirus. Thanks for watching.